Good evening, Vanchika. Okay, I'm Rahul Saha, and in the last class we had talked about how does the sperm provide the phospholipase C zeta, and how does the calcium play an indispensable role in activating the egg, in activating the mRNA present in the ovum, right? And how does the phospholipase C zeta, if not present in the sperm, will lead to improper ICSI, right? Intracytoplasmic sperm injection technique if you want to go for in vitro fertilization. So in today's class, we'll be talking about a very pivotal topic known as cleavage. Okay, blastocyst formation, morula formation, and all that in great detail. Okay, so let's begin our discussion. So what happens is we are well aware of the fact that the the newly formed zygote, I told you it only becomes dinucleated, complete, or you can say it only becomes the diploid nucleus after it has undergone the first mitotic division, right? Before that, the two specific, you can say pronuclei, they do not converse, they do not directly get an amalgamation. So that is also the astral tubules, microtubules are then provided by the, you can say, the centrosome from the sperm itself. So now coming on to this thing, the cleavage and all, we know that mammalian embryogenesis employs some fundamentally different strategies, you can say, from those used by lower vertebrates. Because the placental connection to the mother obviates the need for the developing oocyte to store large amount of yolks. Mammals don't do so because of the placental connection. You can say the eggs of mammals are quite small, and mammalian cleavage is a prolonged process, okay, which typically coincides with the time required to transport the early embryo. Very important. Mammalian cleavage is a prolonged process which requires, you can say, the time, or which requires the same, almost the same amount of time or period of time that is required to transport the early embryo transport the early embryo, you can say, from its site of fertilization in the intrauterine tube, means the ampulla, to the place of implantation in the uterus. Okay, from its site of fertilization to the implantation site. This fertilization occurred in the oviduct, not in the uterus, and that too in the ampullary region of the oviduct, right? So it will be carried from there to the uterus, and then implantation would eventually occur. So this is the time it is also required for cleavage. It just typically coincides with that. Okay. So what happens is a prominent, you can say, innovation in early mammalian embryogenesis. We all know that it is the formation of the trophoblast. Okay. So trophoblast is the specialized tissue We'll go down into the detail of it also. We are all aware of trophoblast, which is the specialized tissue that forms the, you can say, the tropic interface between the embryo and the mother. Okay, specialized tissue from which the placenta develops also. That forms the, you can say, tropic interface between the embryo and the mother. Trophic interface between the embryo and the mother. So that is there. That is the primary work of trophoblast, you can say. Good evening, Tarun. So now what happens is when we talk about cleavage, okay, we go to the morphology of cleavage. So compared with most of other species, okay, mammalian cleavage is a very slow process. You can call it a very leisurely process, okay, which is measured in days rather than hours. Okay, in sea urchin cleavage is of some few hours only. Okay, so mammalian cleavage, okay, comparative level question, takes days. Okay, so it's a leisurely process, you can say, slow process, gradual process. Okay, not, it does not depend on how many hours it will take. It will take some few days. Okay, now the second thing is development proceeds at the rate of roughly one cleavage division. It is that slow, one cleavage division per day for the first 2 days. You draw the diagram also. So it is one cleavage division per day for the first 2 days. Okay, now after the two cell stage is reached, mammalian cleavage becomes asynchronous. Okay, with one of the two cells, okay, dividing to form a 3 cell embryo. Okay, and when the embryo consists of approximately 16 cells, it is called a morula. Okay, I'll draw the diagram. So morula means, it has been derived from a Latin word meaning mulberry. Okay, looks like a mulberry only. So what happens is, you can see in the cleavage, if I draw the diagram, okay, this is the main cell. Okay, first two nuclei is there. Okay, so first A, it is the, you can call it two-celled. Then second day it will become four-celled. That is known as blastomere. Okay, now one, two, three, four. Second day, four cell, essentially four nuclei. Okay, so this is known as blastomere. Now it will keep on growing. Okay, it will keep on dividing. Okay, so it will become 16 cell in 3 days. Okay, in 3 days time it will become 16 cell. Okay, so if you draw a huge one like this, you can put 16 cell, 1 2 3 4 5 6 7 8 9 10 11 12 16. So this is the result of 3 days. Okay, 3 days, 16 celled, known as morula. Okay, it is known as morula. Then 4 days it will become 58 cell blastocyst. Okay, and then the blastocyst will start accumulating fluid in it, that is known as the blastocoele. Okay, and the blastocoele would be having the inner cell mass and the trophoblast both, okay, together. So that is your entirety. Okay, and this is the zona pellucida. Still you have zona pellucida. Okay, this one is the zona pellucida. Don't think it is gone. Still you have zona pellucida. Okay, the outer layer, zona pellucida, still remains. It still remains. So what happens is, you can say that between 8 and 16 cell stages, okay, that is the most important thing that you won't be noticing in any of the content that you've studied till now. Listen to it very, very carefully. Between 8 and 16 cell stages, the embryos of placental mammals, okay, who have placenta, that is the meaning of placental mammals, they enter a phase known as compaction. Okay, what is compaction? It is not, you can say, compression of the chromosome which is activated by or which is perpetuated by protamines when spermatids become spermatozoon. This compaction means which takes place during the individual or during which the individual outer blastomeres, the small cells, nuclei especially, the outer blastomeres tightly adhere through the gap junction and tight junctions and lose their individual identity when viewed from the surface. That is what happens. Okay, so if you have studied extracellular matrix in class 11th, you will be knowing that the occludin and claudin proteins are present to form tight junctions, means you cannot have paracellular transport. Okay, paracellular transport will be completely stopped if you have tight junctions activated between the two cells, because the thing is, cell adhesion is the most important thing in biology, and that plays a dramatic role. Okay, a dramatic role in case of your embryogenesis. So suppose these are the two cells, they need to be adhered. Suppose they are adhered very, very tightly. Why? Suppose this is the occludin protein, occludin claudin. This is again from class 11th. Now this will block the paracellular route of transport. This is known as paracellular route between the two cells. Okay. Through the receptor, some transport is happening and is known as transcellular route, going through the cell. Okay, like gap junctions will be having cell adhesion molecules like integrins, cadherins, selectins. Okay, so those things are present. Now the thing is, they adhere so tightly that the blastomeres tend to overlap. Okay, overlap means from the top if you look, they look like, you can say, half cell this side, half cell this side. They are so tightly adhered that they lose their identity. Means suppose they are like this, almost they look like this. Okay, so they lose their individual identity of becoming an individually independent cell. So they kind of, you can say, overlap. You can say it looks like they are overlapping. So that is the meaning of compaction. So I'll write it down. So between the 8 and 16 cell stages, so these things are heavily asked, extensively asked in your NEET examination. 16 cell stages. The embryos of placental mammals. Of placental mammals. They enter a phase called compaction. So here compaction means during which the individual outer blastomeres, I should be saying the individual outer blastomeres, they adhere tightly through gap and tight junctions, through gap and tight junctions. And what they do is they lose their individual identity when they don't lose it, but it seems like they lose it, okay? Because the adherence is so you can say prolific that they lose their individual identity when viewed from the surface. So always write that line. Their individual identity when viewed from the surface. Okay, so that is why compaction happens. Okay, and again I'll tell you, compaction is also mediated by the concentration of calcium. Okay, because when the calcium influx happens, okay, so calcium activated cell adhesion molecules, they start to express and perform their functions. Number one of those molecules are known as cadherin molecules. Okay, so we have something known as various types of cadherins are present. Okay, number one is the E cadherin. Okay, so we have various types of cadherin molecules. So E cadherin is one of those molecules. Okay, which is known as a cell adhesion molecule. Okay, so E cadherin molecule uses a or forms a ring around the apical surface of the blastomeres. Okay, that is the meaning of E cadherin. E stands for here epithelial cadherin. Okay, so if in NEET examination that question comes, you can say the first molecule to cause E cadherins, they come under CAM. CAM means cell adhesion molecules. Okay. So that is known as epithelial cadherin. Okay. So if we start offline classes, I'll teach you all these things also. Okay, this specific cadherin molecules, selected molecules, integrin molecules, because you wouldn't be knowing the concept regarding it. So that will be a really great help for you when it comes to clearing any kind of competitive examination. So the thing is, it is also activated by calcium influx only. Think how important your calcium is. Okay, so many things that it can be done or it can perpetuate. Now, another factor which could lead to compaction is the contraction of the meshwork of, you can say, a specialized protein known as actomycin. Okay, what happens is there is a specific protein known as actomycin. Okay, now actomycin is a very specific protein present in the blastomere stage. Okay, so actomyosin, when we talk about, it is nothing but a complex of actin and myosin, getting, of which the contractile protein microfilaments are composed. So if you have studied microfilaments, you'll be knowing this actomyosin that I am talking about. Actomyosin. It is nothing but actin plus myosin. Okay. Now it is like a contractile ring which forms a prominent structure during cytokinesis. So come to the cell cycle chapter. Okay. Then only cytokinesis will occur. It forms the contractile ring during cytokinesis, and that is what we are going for in case of cleavage, right? Rapid divisions. So this contractile ring, okay, it leads to the, you can say, contraction of it and therefore quick division. Okay, so the activity of this depends on sodium, potassium, okay, then sodium potassium ATPase transport channel that I told you, okay, and you can say Na plus and water H2O move across the epithelium like outer blastomeres and accumulate in spaces among the inner blastomeres. Since Na plus K plus pump, it keeps on happening, okay, so Na plus and water, okay, also move through the epithelium and they accumulate in the spaces between the inner blastomeres, okay. So this water accumulation occurs approximately 4 days after fertilization, okay. So when this water accumulation is happening, so suppose you have little bit of space here between the blastomeres, this water would come in, and this process, water accumulation, H2O accumulation, okay, is known as, this process which occurs approximately 4 days after fertilization, okay, this occurs 4 days, means after the morula formation. Morula formation happens at 3 days only, okay, or after 3 days only. So this occurs 4 days after fertilization, okay, 4 days after fertilization, and is known as cavitation, okay, cavitation, very very important, is known as cavitation, uptake of, sorry, intake of water into the space between the blastomeres. Now what it will do, it will result in fluid filled space known as the blastocoele or blastocyst cavity, okay, and convert those blastomeres, that you can say morula, into the blastocyst stage. Now it will cause the formation of blastocoele, also referred to as blastocyst cavity, and this is now known as a blastocyst only, from morula to blastocyst, okay. Now at the blastocyst stage, so I will try showing you the blastocyst stage diagram because it will be tough to draw here. Blastocyst stage, if I can show it to you. So just look at this diagram, okay. You have now the blastocoele formation. This blastocoele is what? Accumulation of water and proteins. But main thing is, if you do not accumulate water, you will never be able to form blastocoele, okay, or the blastocyst cavity. Now this entire thing is known as the blastocyst, okay. Now you have the inner cell mass. These cells, these outer cells are known as, the outermost layer is known as trophoblast, and then still it is surrounded by zona pellucida, okay. So the thing is, at the blastocyst stage, the embryo, okay, I have told you, is still surrounded by zona pellucida. Keep in mind always. Okay, I'll write it down. At the blastocyst stage, many of you make the amateur mistake that at blastocyst stage zona pellucida is absent. No, at the blastocyst stage in mammals, we're talking about placental organisms. During the embryogenesis process, the embryo is still surrounded by zona pellucida. Okay, it is still surrounded by zona pellucida. Always know this fact. Okay, and it also consists of two types of cells now. Just I showed you in the diagram. An outer epithelial layer known as the trophoblast, which surrounds the or surrounds a small inner group of cells that is known as inner cell mass. Now, two types of cells are present. Okay, so outer epithelial layer, which are known as the trophoblast and inner cell layer, which is known as an inner cell mass. These are the two sets of cells or two types of cells which are present. Now, each blastomere, okay, at the 2 cell stage, at the 4 cell stage, okay, if you go back to it, each blastomere at the 2 cell stage and 4 cell stage, okay, it contributes cells to both the inner cell mass and trophoblast. Always remember, okay, when the blastomere was 2 cell stage, 4 cell stage, it contributes to the formation of both the inner cell mass. At the each blastomere at the 2 cell stage or you have 4 cell stage at 2 days, it contributes to the formation of both the cell layers. Okay, so keep it in your head perfectly, right? So this is known as the inner cell mass and trophoblast. Now the end of the blastocyst, okay, that contains the inner cell mass. Always note this is very seldom given in the books, but it's asked and must be known conceptually also. The end of the blastocyst that contains the inner cell mass is known as the embryonic pole, and the opposite end is known as the aembryonic pole or the vegetal pole. Okay, very clearly I'm telling you, that is the end of the, I'll show it to you on the image also. I'll first write it down. The end of the blastocyst that contains the inner cell mass is known as embryonic pole. And the opposite end is known as the aembryonic pole. So embryonic pole is also known as animal pole. Okay, sometimes aembryonic pole is also known as vegetal pole. Okay, in few cases. The thing is, this side where it contains the inner cell mass, this side is known as the animal pole or embryonic pole. This side, the polarity is, it is known by the name as vegetal pole or aembryonic pole. Okay, because from this side, the morphogenesis of the fetus or of the embryo will not occur and it will not form fetus. Okay, so that is known as embryonic pole and vegetal pole. So that is the concept there. Now, the appearance of the two cell types, it reflects major organizational changes, okay, that have occurred within the embryo, and it also represents the specialization of the blastomeres into two distinct cell lineages, because these two are the different cell types. Okay, so you can call them as, if you go for, if you have studied the hematopoiesis from the hematopoietic stem cells present in the red bone marrow, they go for myeloid cell division and lymphoid cell, then various cells like megakaryocytes, then you have erythrocytes, okay, then different leukocytes, then this side lymphocytes, the, you can say B cells, T cells, actually killer cells, monocytes, macrophages are developed, right? So those are all different lineages. First you have myeloid lineage and lymphoid, you can say progenitor cell lineage. This is also two different cell type completely, two different cell lineage, you can say. Cell type or cell lineage, they take two different pathways and become two different cells. One is the inner cell mass, one is the trophoblast. Okay. Now, if we come to the control, the molecular dynamics of the early cleavage, okay, so along with the increase in cell number, you can say the mammalian cleavage is a period, you can say, which is dominated by various types of critical developmental events. Okay. Now, the earliest event which happens in the cleavage division or during the cleavage division is, you can say, the transition from maternally to zygotically produced gene products. Now, the gene product expression changes not maternally but zygotically. You can say dominated gene products are produced. Number one, you can say during cleavage, shift to zygotically dominant gene product formation. That is the number one. Second is the compaction that I talked about. Okay. Compaction means what? During which the blastomeres they will develop intercellular connections and begin to take on characteristics of epithelial cells, right? That is why I told you you have blasts are known as epithelial cells, right? They resulted from the blastomeres only. Okay, so intercellular connections are formed via gap and tight junctions, and the blastomeres, you can say, they begin to take characteristics of epithelial cells, and this also forms between the blastomeres. Only intercellular connections begin to take on, or you can say take up, the characteristics of the epithelial cell type. Okay, now what happens is, the third event which happens is the polarization of individual blastomeres. Okay, this sets the stage for the developmental decision that results in the, you can say, subdivision of the embryo undergoing cleavage into two distinct types of cells, the trophoblast and the inner cell mass. Okay, that is known as polarization. Distinct pathway pertaining to two lineages, means cell types, forming trophoblast, outer epithelial cell layer, and inner cell mass, inner cell layer, that is known as polarization, and then only your vegetal pole and animal pole or your embryonic and aembryonic pole will be started to or will be commencing to produce. Now, because you can say of the lack of massive storage of maternal ribosomes and RNAs during oogenesis, okay, development of the mammalian embryo must rely on the activation of zygotic gene products at a very early stage. Okay, so most maternal transcription products are degraded by the two cell stage. Okay, because they do store ribosomes and mRNA, but not for a very long period of time. Okay, so they store it for a very little period of time, that is still the two cell stage reliance only. So a lot of maternal ribosomes and RNAs, you can say a huge amount is not stored. Okay, so you can say due to lack of maternal RNAs and ribosomes, the zygote gene product formation becomes significant. Means that I told you after the two cell stage only, okay, after the two cell stage only maternal transcription products are degraded. Okay, so maternal transcription products are degraded after two cell stage only. So now what happens is this will activate the or this will stimulate the activation of the embryonic genome, okay, which begins producing mRNAs from a or tRNAs, RNAs, all types of rRNAs from a significant number of genes, okay, like more than 1500 genes will start producing RNAs by the time cleavage has advanced to the 4 cell stage. Okay, now they have reached a very advanced stage. Many genes are producing RNAs, you can say. So you can say that there does not seem to be a sharp transition from the cessation of reliance on, you can say, purely maternal gene products and the initiation of transcription from the embryonic genome. Okay, so we have multiple gene products getting produced, okay, which is not in your course, like beta 2 microglobulin, you have beta glucuronidase. So these are not in your course, but they appear in the embryo very early. Okay, while maternal actin and histone mRNAs, okay, are still being used. Okay, they are still being used for production of proteins. But the thing is, how much the early embryo relies on its own gene product, okay, like development past the two cell stage does not occur in mouse if mRNA transcription is inhibited. But the thing is, you can say that mature eggs and sperms, okay, are transcriptionally inactive. Okay, mature eggs and sperms, very important thing this is, they are transcriptionally inactive. I'm not talking about the zygote, I'm talking about the egg and sperm. What is the reason for this? Their DNA is highly methylated, okay, at cytosine and guanine. They are known as CpG island, like followed by, suppose you have this kind of, so CG like this. So they are known as CpG island methylation at cytosine and at guanine. Okay, so mature egg and sperm, they are transcriptionally inactive because they don't want to spent energy they have already acquired, okay, during the oogenesis, spermatogenesis, spermatogenesis process. They have accumulated whatever is necessary. Now they don't want to spend that much energy in transcribing, because that also requires NTPs, right? So that is utilization of ATP only, each with each, you can say, nucleotide or ribonucleotide being added. So they are transcriptionally inactive due to methylation of the genome, as RNA polymerase cannot come and bind due to steric hindrance caused by methylation of the genome of the DNA at CPG islands. Okay. So methylation is able to activate, okay, this gene expression, that is also known as epigenetic modification, right? Now, within 4 hours after fertilization, okay, the paternally derived genome, which is given by the sperm, it goes rapid demethylation, okay, and demethylation of the paternally derived genome occurs more gradually, okay, until the early morula, at which stage all the DNA is demethylated. Okay, so maternally is also demethylated, paternally is also demethylated. But the thing is, paternal demethylation takes more time. But since the or after the arrival at the stage of morula, you can say all the paternal DNA is maximally demethylated. So that is the event, and then it leads to zygotic gene products. Okay, the zygotic gene product formation. Okay, now there is something known as paternal imprinting also. Now it has shown that in some experiments that the expression of certain genes which are derived from the egg, because both the egg and sperm, they contain the same genes, they're just the, you can say, different alleles, homologous pairs. So the expression of certain genes derived from the egg differs from the expression of the same genes derived from the spermatozoon. That also happens. Okay, so suppose chromosome number 1 of egg, chromosome number 1 of sperm, at the same locus you have gene 1, you have gene 1. The expression of both the genes, expression means how much mRNA is produced from both the genes, will differ. This process is known as paternal imprinting. Paternal imprinting, very, very important process. Okay, so if a male or female pronucleus is removed, okay, and replaced with a corresponding male or female pronucleus, you can say development is normal. But if a male pronucleus is removed and replaced with a female pronucleus, have you ever thought of this experiment? That in the end, I am providing you only the DNA, the sperm provides DNA and the centrioles. So just swap that DNA with the DNA got from the another ovum. Again, that will have 23 chromosomes only. So how will it know, the zygote know which is which DNA? That is it this DNA from sperm or ovum? How will it know? So suppose that experiment I do, great experiment that is, that a male pronucleus and female pronucleus, okay, in that case if a male pronucleus is removed and replaced with a female pronucleus, okay, resulting in, suppose a male pronucleus is removed, okay, and replaced by a female replaced by a female pronucleus. that would lead to the, you can say, zygote having two female pronuclei or not, two female pronuclei. And experimentally, what has been seen is the embryo develops fairly normally, but the placenta and yolk sac are very, very poorly developed. Okay, here in this case, the two female pronuclei, the development of embryo is almost normal, but placenta and yolk sac development is quite poor, so you cannot sustain with the pregnancy. Okay, yolk sac development is poor in this case. Now, second case is, you can say that if a zygote with two male pronuclei is put, here two female was there or were there, now if you have a zygote with two male pronuclei, means if you take out that one female pronuclei and replace it with a male pronuclei, then what will happen? Here the embryo growth is stunted. Okay, embryo development, just the opposite, stunted or abnormal, but the placenta and yolk sac development remains fairly normal. Almost normal. Now, what does it mean? That it is the epigenetics, the imprinting is epigenetic, that how much of the genes are expressed. So if female and male, both the genes are expressed differently, if you put two female, the expression will be the same, and that will lead to the yolk and placenta being, you can say, developed in a very, very stunted way. Same if you put two females, again the expression will coincide and differential expression will be removed or will be truncated, and then the embryo development will be stunted. Okay, so that is how, and that is how intricately poised the development is. You cannot just fool the nature, because you will think that no, this is only DNA, no problem, and we will proceed with the process. So that will not happen, okay, at all, at any cost. So in case of females, also one question comes in NEET examination regarding X inactivation. Okay, so you have two X chromosomes. Okay, that we had talked about in the NEET examination also. You have two X chromosomes, so you will X chromosomes. It not only contains the secondary sex character, you can say associated genes, but a lot of other genes like SARS-CoV-2 binds to ACE2. So this ACE2 angiotensin receptor, angiotensin or angiotensinogen converting enzyme ACE type 2 is present. That gene is present on X chromosome only. Okay, so you don't want double expression, because males have only one X chromosome, then your body will be, your physiology of the female body will be extremely different. You won't be able to take the same medicine. Okay, then the two sexes will have completely different physiology. So what the body does is it creates a bare body and will, you can say, inactivate, epigenetically silence, that is called, epigenetically silence one of the X chromosomes. Okay, that is random which X chromosome will be silenced. Okay, so X chromosome inactivation is initiated where? At the X inactivation center. Okay, which is a unique locus on the X chromosome, that is known as X inactivation center. Suppose this is the X inactivation center, then formation of bare body happens. Now there is something known as XIST, X inactive specific transcript. Okay, X inactive specific transcript, very, very important. XIST, X inactive specific transcript. Now this transcript does what? It produces a large RNA with no protein coding potential. Okay. Now cyst RNA remains in the nucleus only. Okay. It produces a large RNA with no protein coding ability or potential. Now what does it do is this cyst RNA remains in the nucleus and coats the entire inactive X chromosome. Coats the entire inactive X chromosome. So whenever that question comes in NEET examination, you should tick on cyst causes X chromosome inactivation. Cyst coats the inactive X chromosome. So these two questions can be asked, okay, in NEET examination. So it does not allow any further transcription from that chromosome due to again steric hindrance. Okay. Now in the inactivated X chromosome, the cyst gene is unmethylated and expressed so that RNA can be produced. Okay. And in the active X chromosome, the cyst gene will be methylated and silent. Very, very important, you can say, demarcation. Active X chromosome, this is inactive X chromosome. So for inactivating the X chromosome, you need cyst RNA or not? You need cyst RNA. So the you can say the gene coding for X inactive specific transcript, the gene here also cyst gene. So for this one, you don't need the cyst RNA to be produced. Okay, you don't want the RNA polymerase to bind to the promoter of the cyst gene. So what you will do is, or what the cell will do is, randomly it will choose, that research is still going on, which chromosome is chosen and why, which of the two X chromosomes is chosen and why. But till now, randomly one X chromosome will be chosen, and the cyst gene of that chromosome or that X chromosome will be epigenetically silent due to, again, just like I told you in case of sperm and ovum, DNA is heavily methylated. Okay, or you can call it extensively methylated. Here the cyst gene will be epigenetically active because you want the cyst RNA to be transcribed so that it can bind and inactivate the X chromosome. This is inactivated X chromosome, right? Okay, epigenetically active means non-silent or not silent. Then it means it is extensively, the DNA is extensively unmethylated. Now it will be produced and it will coat that X chromosome specifically from which it is produced. That entire process is there. Okay, that same chromosome it will coat and then cause inactivation of that X chromosome. Okay, so we have learned the cleavage. Okay, we have learned how the cleavage occurs. Okay, and also the thing is, in the next lecture we'll be going for, okay, the formation of the germ layers, okay, ectoderm, mesoderm, endoderm, also the syncytiotropoblast layer, okay, from the how does placenta act as endocrine gland and how does it get regulated, and then we'll be switching on to the menstrual cycle and the female reproductive tract. So I hope you are able to comprehend the lecture. So if you had any kind of queries or doubts, you can definitely contact me on my personal number, and I'll be replying as soon as